ETM Hotel Rennie Sean, welcome in peace. My name is Sean. I'd like to thank everybody for tuning into this episode of Science with Sean. Again, man, if you're a part of our virology class and lecture series, it's study group time. It's study group time. I don't expect a lot of people to be involved this evening. They may be distracted by the noise, but it's a good opportunity for me to study. So while people are caught up in the emotionalisms of their own ignorances, I think this is a good opportunity for me to get some reading, some learning, some researching, all of those things that make us more useful for our people in. That's not a shot or a slight at anyone in particular. However, I just see these as opportunities for me to develop and grow and move the information forward. So while some people will stay stuck, we'll continue to progress. If you haven't picked up your copy of what Sars Cov2 taught me, the link is in the description. Also, if you have purchased your copy, there'll be sometime this coming months we will get into the book in particular, and we'll deal with what we need to deal with from the perspective to make sure that we're all growing and learning. Hopefully, I sound a little bit clear and a little bit better than a second ago. I do want to say peace to anyone that's viewing this right now. I do see we got Brother Keenan, Teddy, and Omar in the chat. Um, I want to apologize for pulling y'all off any streams that I may have pulled y'all off of, but uh, man, I appreciate y'all sliding in, man, and rocking out with me while we handle our business on this virology tip. So today's study group is centered around reverse transcription and integration. Uh, man, it's a lot of work been put in today. What is this video? This is the fourth video. So we're not really going to spend a lot of time on it. We're going to section it up maybe tomorrow evening or Sunday evening. I come back Sunday evening and we do the part 2 and we wrestle through it, but we get ready for Monday and Wednesdays series, really Mondays. That'll help us be able to do what we need to do to get things going. So with that being said, I do want to go ahead and talk about reverse transcriptions and integrations, and then we'll get into the lecture and we'll learn as we go. I feel like if we're prepared for the week, we have a better outcome. So here we go. Retroviral reverse transcriptions. There's a quote that actually is used in the book that says, "One can't believe impossible things," said Alice. "I dare you to say you haven't had much practice," said the Queen. "While sometimes I've believed as many as 6 impossible things before breakfast." That's a quote from Lewis Carroll and the Alice of Wonderland. I don't know why that was in there, but it's some corny ha-ha stuff. But back-to-back reports from laboratories of Howard Timmon and David Baltimore in 1970 provided the first concrete evidence for the presence of RNA direct DNA polymerase activity in retro particles. This is chapter 10 of the book, so I don't know what chapter it may be in a later series or a different edition, but it's reverse transcriptions and integrations. In Timmons' case, the discovery came about throughout attempts to understand how infection by members of this group of viruses, which have plus-strand RNA genomes, could alter the heredity of cells permanently, as they do in process of uncognate transformation. Let's look up what the word uncognate means and see if we get a generalization of the word. Unfortunately, I have the physical copy of the book and I can't share with you what I'm looking at. So I'm going to do my best job to try to slow things down and have you walk with me so that we have an understanding before we even listen to Vince get into whatever he want to get into. So, uncognate, oncogenetic transformation is a multi-stage process of successive acquisitions of genetic and epigenetic alterations affecting cell proliferations and survival. All right, with a few notable exceptions, different stages of cancer regressions are not easily distinguishable and therefore not clearly defined. Let's see if we can get a oncogenic transformation for dummies. They ain't rocking with us on a on a dumbed down definition. And then there's a long video here, and this by Vincent Racaniello. Let's see if there's any short, like 2 minutes. Transformation of normal cells and cancers, mechanisms, and therefore. All right, so let's continue because we're not going to find a clear-cut definition that we actually need to get into the actual lecture. All right, so let me find my place. Studies of bacterial viruses such as bacteriophage lambda had established a precedent for viral DNA integration into host DNA. Timmon believed that retroviral RNA genomes became integrated into the host cell chromatome in a DNA form. This seemed an outlandish idea at the time because it says this seemed as, it should have been as an outlandish idea, but these are virologists, they're not grammar students. Could copy, at a time, virus polymers could copy DNA and DNA templates, RNA and RNA templates. Furthermore, it was difficult hypothesis to test with technology then available at attempts by Timmon and others to demonstrate retroviral DNA is made from genetic RNA in effective cells were generally met with skepticism. Baltimore's entree into problem of reverse transcription came from his interest in the nucleic acid polymerizing enzymes, right? Recently discovered within the particles of certain viruses. It occurred to Baltimore and Timmon independently that retrovirus particles might also include an enzyme. In this case, the sought-after RNA-dependent DNA polymers. Subsequent experiments confirmed this prediction. The enzymes that had earlier eluded Timmon discovered to be an integral component of the virus particles. 5 years later, Timmon and Baltimore were rewarded Nobel Prizes for physiology of medicine for their independent discoveries of retroviral reverse transcription in avian route saccharomyces viruses and leukemia virus particles. So here we have retroviral transcriptus. And a Nobel Prize was awarded to Timmon in Baltimore. David Baltimore in 1970, Howard Timmon 1970. Now the impact was immediate of the discoveries of RT. And when we say RT, we're talking reverse transcription. Was to amend the central dogma of molecular biology that the transfer of genetic information is unidirectional DNA, RNA, protein. It was now apparent that there could be also retrograde flow of information from RNA to DNA, and the name retroviruses eventually came to replace the earlier designation of RNA tumor viruses. As Timmon hypothesized, study of oncogenetic retroviruses had provided a framework for current concepts of the genetic basis of cancer. Analysis of the reverse transcription and integration processes has enhanced our understanding of how retroviral infections persisted and has clarified aspects of pathogenesis of acquired immunodeficiency syndrome, AIDS, caused by human immunodeficiency virus type 1, HIV. Finally, RT itself, first purified from virus particles and now produced in bacterium, has become an indispensable tool in molecular biology, allowing experimentalists to capture cellular messenger RNA, complementary DNA, cDNA, that's what they call cDNA, complementary DNA, which can be converted into double-stranded DNA, cloned and expressed by now well-established methodologies. Excuse me. Reverse transcription followed by amplification of DNA product via polymerase chain reaction, RT-PCR, is the most sensitive technique for the detection and quantification of cellular viral RNAs in the lab and the clinic. Furthermore, high deficiency of DNA integration mediated by retroviral RT partner enzyme, integrase, has been widely exploited in construction of viral vectors for gene transfer. For such reasons, they devoted an entire chapter to this. So, reverse transcriptions and integrations. Reverse transcriptions, which is RTs, are enzymes that synthesize DNA from both RNA and DNA templates. One immediate consequence of discovery of first retroviral RT was the amendment of the central dogma DNA, RNA, and protein. The double-stranded DNA product of retroviral reverse transcription is collinear with the viral RNA genome, but includes long terminal repeats, or what they call LTRs. LTRs is long terminal repeats, containing signals required for its integration and subsequent transcription by host cell proteins. Retroviruses are the only animal viruses that encode a specialized recombinant integrase, which, or what they call IN, which mediates the insections of the viral into the host genome, where it is called a provirus. Retroviral is an indispensable research and clinical tool. Retrotransposons are elements in cellular DNA that are copied from an RNA immediate by RTs and inserted other loci. Such intracellular transposal elements are present in the genomes of most, if not all, members of the tree of life. RT and IN, remember RT is reverse transcription, IT is integrase. All right. Like enzymes, probably arose early in evolution before the appearance of plants, animals, and their viruses. Viruses that depend on such enzymes for their reproduction are widespread in animals, plants, and fungi. As with many DNA polymers, DNA synthesis by RT requires a primer, either a fragment of RNA or protein. Reverse transcription is an error-prone process at RT's lack of proofreading activity. Reverse transcription of retroviral or hypnoviral RNAs is facilitated by the presence of terminal repeat sequences and the dynamic of multifunctional properties of the RTs. Genomic RNA templates are packaged by all reverse transcriptional viruses, but the RTs of some of these viruses convert RNA to DNA copied before infection of a new cell. Now, I read you some of the highlighted versions of this particular chapter, and let's see what we get into with Vincent Racaniello.
Of seemingly impossible things to talk about today. This is a very cool subject. This story begins in 1908. A little bit of history about tumor viruses. 1908, Bang and Ellerman discovered chicken leukemia virus. Remember, viruses were just discovered at the end of the 1800s, and we were just understanding filterable viruses, and so people were taking agents and putting them through filters, and they found that chicken leukemia seemed to be caused by a filterable virus. Now, leukemia at the time wasn't considered to be a cancer, wasn't a solid tumor, which of course is wrong. It is a cancer, but it wasn't recognized as the first cancer virus until many years later. In 1911, Peyton Rouse discovered Rouse sarcoma virus. This is a virus that causes solid tumors in chickens, and we will talk about that in more detail when we talk about transformation and oncogenesis. He didn't get the Nobel Prize till 55 years later. It took a long time for it to be recognized that what he did was important, and the longest incubation period for a Nobel Prize. At some point after this, they were called tumor viruses, and many years later, they were in fact found to have RNA genomes, and so we call them now RNA tumor viruses, and that's to distinguish them from DNA tumor viruses, which we will also talk about later. Now, a key part of the history of RNA tumor viruses is provided by Howard Temin. So as you will see when we talk about transformation and oncogenesis, Temin took Rouse sarcoma virus and infected chicken cells and found that the virus induced permanent changes in the chicken cells. So, wow, how can an RNA virus do that, he thought? Well, it's because he thought, anyway, that the virus RNA must be changed into DNA. And so he came up with the provirus hypothesis, the idea that somehow the RNA tumor virus genome is converted to DNA and becomes a permanent part of the DNA. And this is the provirus hypothesis. This is the idea that the tumor virus RNA gets copied to DNA and then gets integrated into the host DNA. And that's what the provirus is. Now, David Baltimore had heard this theory from Temin, and he had been working on RNA polymerases. Remember, he made up the Baltimore scheme, and he had come up with the idea that plus-strand RNA viruses, because they're mRNA, they don't have to have RNA polymerase in the particle. On the other hand, the minus-strand RNA viruses have to have RNA polymerase in the particle because the cell can't copy RNA of an RNA virus. So he thought these RNA tumor viruses must have an enzyme in the particle to copy the RNA to DNA. That must be a virus-associated enzyme, because he thought, well, the cells can't do that. He was right, although, as you will see, cells do have reverse transcriptase, the enzyme that we're talking about today. So he and Temin both worked on this problem, and in a very short time, they both discovered independently reverse transcriptase in RNA tumor virus particles. And that was published back-to-back in Nature in 1975. And let me just take a pause here, and I want to look at the chat because I want to make sure that everything is going well. Okay, everybody see and hear everything okay, right? I'm at Columbia today, which is where I used to do most of my pods, and with the wonderful background, with all the cool stuff, which we don't have at the incubator, but oh well. Okay, everything seems to be good. Nobody's complaining that they can't hear me, so let's resume. These two papers came out in Nature, 1975, RNA-dependent RNA polymerase in virions of Rouse sarcoma virus from Temin, and RNA-dependent DNA polymerase in virions of RNA tumor viruses from Baltimore. And these were my two picks for seminal papers on TWIV not too long ago, last week. And so this enzyme was called reverse transcriptase. And Yes, I did do a TWIV with David Baltimore. Actually, he's been on TWIV twice, and I think I also interviewed him separately. So, yeah, I've talked to him a lot. Well, he was my postdoc advisor, of course, so I have a special in, right? So the enzyme is reverse transcriptase. And the viruses now were called retroviruses. We switched from RNA tumor virus to retroviruses because of the ability to reverse the flow of genetic information. As you know, Crick originally postulated the flow of information goes from DNA to RNA to protein, and retroviruses at least are able to reverse part of this. They go from RNA to DNA, and then theoretically they would go back to RNA and to protein. So that's where the name of the viruses come from. And reverse transcriptase is to make the DNA copy of RNA. Reverse transcriptase is an amazing discovery. It's an amazing enzyme. It has revolutionized molecular biology, right? Not just in the lab. I mean, you can buy this stuff now readily. When I was working with RT in the late 70s, you couldn't buy it. So we found people who made it and got them to donate it to us. But it has been indispensable for research. It has been indispensable for biotechnology. It has been indispensable for diagnosing infections. Every Sars Cov2 PCR test utilizes RT. That's where RT comes from in the name, RT-PCR, reverse transcriptase PCR. You use RT to make a DNA copy of the RNA, and then you amplify the DNA with PCR. This is just an amazing discovery. And neither discoverer patented it. It's really remarkable. In fact, one of the interviews I did with David, he reminisced and he said, maybe I should have patented it. But they didn't think of it at the time. You know, now modern days, everyone's patenting everything right away. And back then, they just discovered things for the good of humanity. So today we're going to talk about two families of viruses with reverse transcriptase in their reproduction cycles. We're going to talk about the retroviruses, of course, which have a plus-stranded RNA genome that gets converted first to negative sense DNA, and then that's made double-stranded. And of course, the DNA is transcribed to mRNA in order to make proteins. And yes, an enzyme can certainly be patented, as well as applications derived from its use. Then we're going to talk about hepatitis B virus, which has a DNA genome, an unusual one, a gapped DNA genome, but it has reverse transcriptase in its reproduction cycle. It's not shown here, but we'll go into that. But first we do retroviruses. And we're going to start with Rous sarcoma virus, which is a retrovirus. And it's an envelope virus with glycoproteins in the envelope, as you can see here. Inside there is a icosahedral nucleocapsid, and the plus-strand RNA is here in two copies. Interesting. Two copies of the viral RNA here. And they're coated with a protein, the nucleocapsid protein. So this is a nucleocapsid. Also in the particle is the reverse transcriptase, and also a few other enzymes, the protease, which is needed to mature the particle after it buds from the cell. We'll talk about that, I believe, on Monday. And then the integrase, which integrates the DNA copy of the viral RNA into the host cell genome. So that's the morphology. Here's an electron micrograph. You can see the nucleocapsid dense core in the middle and the envelope. Retroviruses we classify broadly into retroviruses with simple genomes or with complex genomes. And the Sprouse sarcoma-like viruses that we will talk about today are retroviruses with simple genomes. Here's a diagram of proviral DNA. So again, here's the definition. Proviral DNA is retroviral DNA integrated into the host cell genome. Very specific definition. And you see it's flanked by what are called LTRs. And I know many of you don't like all these abbreviations. This stands for long terminal repeat. You have to abbreviate. It would have fit, right?
I remember, and what I read, Anusha, I think you were late, but I read what LTRs are, long terminal repeats. IN is integrase, dealing with integrase. RTs are reverse transcriptions. Those are the first three abbreviations that he's really dealing with, or the main three that he's dealing with in this lecture. RTs, retrovirus. LTRs are long terminal repeats. Integrase has to deal with INs. He don't even, well, ER would be endogamous reactions. But you know, that's not present, but we do see RT, we do see IN, and we do see LTRs.
And then there are three open reading frames encoding proteins. Here we have the GAG protein, we have the POL, the polymerase, the reverse transcriptase, and the envelope, which is the spike in the membrane of the virus, right? So GAG stands for Group Specific Antigen. It was named years ago when proteins were detected using antibodies only. And those make up the structural proteins of the virus, except for the envelope. This provirus, integrated into the host cell DNA, of course, is transcribed by host cell Pol2 to make an mRNA. All right, there's the mRNA. And so there are two forms of the mRNA. There's an unspliced form here, which is what is going to be incorporated into new virus particles. It encodes GAG, POL, and envelope. It is translated. The translation gives you either GAG alone, or sometimes the stop codon after GAG is suppressed, and you get a longer GAG-POL precursor from which the reverse transcriptase and integrase are produced by proteolytic cleavage. So that's how you access the GAG and the POL open reading frames. And then the envelope protein is produced from a spliced mRNA, shown at the bottom here. But remember, you need to get some of this unspliced RNA out of the nucleus in order to access the GAG and the POL genes, but also to package it into virus particles. And we talked about how that happens, the trick that retroviruses utilize to do that. A little overview of the reproduction cycle. We have virus attaching to cell surface receptors. Many of these retroviruses, the membrane of the virus fuses with that of the plasma membrane and deposits the nucleocapsid in the cytoplasm. This nucleocapsid is permeable, so NTPs can get in. And in fact, that's where reverse transcription begins. Right in the cytoplasm, you get the DNA copy of the RNA made, double-stranded DNA. That then goes into the nucleus of the cell. The DNA integrates into the host cell DNA, and there it's transcribed by Pol2 to make mRNAs, which then, of course, go out in the cytoplasm and are translated to proteins. We will talk about assembly on Monday. Reverse transcriptase is an enzyme that needs a primer. It can be DNA or RNA, though. The primer can be either DNA or RNA, and you'll see how that works out when we talk about reverse transcription in a moment. The template can also be RNA or DNA. Of course, it's going to copy viral RNA into a DNA, right? But then it needs to make the DNA double-stranded. So it can use both RNA and DNA templates. But it only incorporates dNTPs, not rNTPs. It makes DNA, it does not make RNA, no matter what the primer.
What is dNTPs? Anybody know what he's talking about when he say dNTPs? DNA transcriptions, RNA transcriptions that sound familiar.
Primer and template. Reverse transcriptase is an ancient enzyme. Bacteria, archaea, and eukaryotes all have reverse transcriptase activity in the cells, independent of retroviruses. So RT existed long before retroviruses, and it came from the cell. Here's a phylogenetic tree of life to show you bacteria, archaea, eukaryotes. They all derive from a common ancestor, and we think it's likely that the common ancestor had reverse transcriptase. And that's how all three domains of life received reverse transcriptase. Reverse transcriptase evolved before the separation of these three domains of life. And in fact, we think RT might have been the bridge between the RNA world and the modern DNA world. So remember, an RNA world is thought to have existed before DNA-based organisms arose. And at some point in that RNA world, proteins arose. And among those proteins was reverse transcriptase, which then made DNA from the RNA, and then the DNA world took off. So this is a circular phylogenetic tree of all the reverse transcriptases that we know about. The ones in blue are viral or virus-like reverse transcriptases here. The ones in orange are bacterial and archaeal. And then the rest are cellular in various kinds of cells. We'll talk about that in a bit. So RT is present in cells, but it turns out it's very low amounts, so it's not enough to reproduce a retrovirus. So that's why the retroviruses have to have the enzyme in the particle. We also see not only reverse transcriptase in retroviruses, but in hepatitis B virus and in Caulimoviridae, which is a family of viruses that infects plants, cauliflowers, and it has reverse transcriptase. We'll touch on that in a bit. So the reverse transcriptase protein is shown as a box here, right? RNA-dependent DNA polymerase. We've talked a lot about RNA-dependent RNA polymerases. We've talked about DNA-dependent DNA polymerases. We've talked about DNA-dependent RNA polymerases, each in separate sessions. Today, RT, and it has homology at the protein, at the amino acid level with these other polymerases, as you can see by the colored boxes. It has an active site comprising this yellow part here, which here's the RT from HIV. You can see, again, it's a right hand, palm and fingers domain, an active site. Thumb, fingers, and palm, the active site in the palm. And the active site has conserved amino acids, as do the other polymerases. Here for the HIV reverse transcriptase, it's MET, ASP, ASP, but of course the two ASPs are involved in coordinating the magnesium. Now, RNA reverse transcriptase has a second enzymatic activity. It doesn't just copy RNA into DNA. It has another enzyme called RNase H. And RNase H is an enzyme that cleaves RNA, but only when it's double-stranded, which I call duplex form. It could be RNA-RNA, or it could be RNA-DNA. And what it does is make endonucleolytic cleavages. That means it cuts internally, not at the ends. If an enzyme cuts from the ends of a nucleic acid, that would be an exonuclease. So RNase H is an endonuclease, and these red arrows showing it cutting, in this case, an RNA-DNA hybrid. And it makes oligonucleotides, which is a short piece of nucleic acid with a 5' phosphate and a 3' hydroxyl. So it cuts between the oxygen and the phosphate between each base, and you get short oligonucleotides. Now, what's the purpose of this? You'll see. It's essentially to get rid of the RNA template, because once it's copied into DNA, it's no longer needed. Here's the structure of HIV-1 reverse transcriptase. This is actually a dimer of two proteins, 66 and
You have to turn the page at 349.
Dalton subunits. And you can see their contributions to the final enzyme because of the coloring. So here in orange, this forms part of the RNase H activity. And so the green is the thumb domain, the fingers are the blue, and the active site is here in yellow. Not well, all the yellow isn't all the active. It's more than the active site. The active site is actually up here in the palm domain. And here we see a molecule of RNA that has entered the enzyme active site, and it's been copied to DNA. So what's coming out of the active site is a DNA RNA duplex. All right, so the left part of this RT has the reverse transcriptase activity, and the right part has RNase H. So here's a cartoon that explains how this works.
Mm-hmm.
And, you know, by the way, so someone asked, do these have colors at the molecular level? Of course not, they don't. We use the colors. We use the colors for clarity.
The next slide is 337, which you'll see the phyllo tree
class
resemblance.
About viruses. And I showed them a picture of viruses with different colors. And one kid just spontaneously asked me, do you color them so you can tell them apart? Bingo, brilliant kid. Probably not going to be a scientist, though. Probably be a lawyer or a banker or something like that. But yeah, we color them so that we can trace parts of proteins and so forth, but they don't have color at that level. So here's a cartoon of the reverse transcriptase. So here is the polymerase active site right here. So the RNA comes in from the left. There are the two metals, the two magnesium ions that are coordinated by the aspartate residues to help add the bases, fingers and thumb domain. The RNA passes through, and a DNA is made complementary to it. So now you see a duplex DNA RNA passing past the active site, and then it encounters the RNase H site here on the right, shown in brown, and that site also has metals, and that reverses the polymerization. You need metals to do that as well, and the RNA is degraded. So what comes out is a single-stranded DNA. And eventually that will be copied by another polymerase to make it double-stranded. The same one is not able to do both. So RNA in, single-stranded DNA out. That's the function of the RNase H. Overall DNA synthesis is very slow. It takes 4 hours to copy a single 9 kilobase viral RNA. That's the size of the RNA tumor virus.
So again, in the model for DNA RNA hybrid bound to HIV-1, which he's using here in reverse transcription, the RNA template DNA product duplex is shown lying in a cleft. The polymerase active site and the putative RNase H active site. Do y'all see where it's R and then NAS? It's at the bottom. It says RNase H active site are indicated. The ME2 plus, which is near the green area, or if you go to the left where the double-stranded DNA lies, most people, if you're not paying attention, ladies and gentlemen, this green and this blue is representing DNA, right? Is it? It's saying DNA out right here. DNA. All right, here you go. You got the whole thing going on. So anyway, what he's saying here is the DNTP. Y'all see DNTP? Yes, no, maybe so. No, probably not. All right. Which represents deoxide ribonucleotide triphosphate. Substrand interacts directly with two fingertip residues, which induces closure of the binding pocket. The conformational change facilitates attacks at the 3' OH mark of the primer on the X phosphate at the DNTP. All right. Releases of the diphosphate product, addition of the resulting deoxyribonucleotide monophosphate, DNMP, which is at the top next to thumb, and reopening the fingers allows the template primer to translocate and one nucleotide in preparation of the binding to the next DNTP. I could elaborate more, but let them go. We finna take a quiz.
And also very error prone. It makes a mistake every 10,000 to a million bases polymerase, depending on the polymerase. A mistake every 10,000 bases. So that means every time this 9 kilobase genome is copied, one error is introduced. Really, really remarkable. Okie dokie, time for a quiz. Let me go to...
Told y'all. Quiz like a mug. So, y'all know how this go, man. Let's see if we learned anything. Reverse transcriptists or just say reverse transcriptions, have revolutionized molecular biology. Which statement about enzymes is not correct? RT is unique to retroviruses. RT is packaged in the retrovirus particle. The RT protein also has RNase H activity. The name of the enzyme comes from its ability to reverse the flow of genetic information, might have bridged the ancient RNA world with the DNA world. Which statement about the enzyme is not correct? Study long, study wrong. You want me to go over this thing again real quick? RTs are enzymes that synthesize DNA from both RNA and DNA templates. One immediate consequence of the discovery of the first RT was amended in the central dogma DNA RNA protein. Double-stranded DNA product of retroviral reverse transcription is collinear in the viral RNA genome, but includes LTRs containing signals required for its integration and subsequent transcription by host cell proteins. Retroviruses are the only animal viruses that encode a specialized recombinant integrase, which mediates the intersection of the viral DNA into the host genome, where it is called a provirus. Retroviral RT is indispensable research in the clinical tool. Retrotransposons are elements in cellular DNA that are copied from RNA mediated by RTs and inserted at the other loci. Let me deal with a term that you may not have caught wind of. The RNase H or RT also requires a divalent cation, like RNase H enzymes present all bacteria, archaeal, and eukaryotic cells. You heard them say that. So RT is unique with retroviruses. RT is packaged in the retrovirus particle. The name of the enzyme comes from its ability to reverse flow of genetic information. Might have bridged the ancient RNA world with the DNA world. I'm going with what you going with. Oh, y'all wrong. Don't jump out the window. Look at what makes sense and what does not make sense. He's talking about reverse transcription, yes. Again, he's talking about reverse transcriptions, yes. But think about the choices. What? Think about the reverse transcriptase has revolutionized molecular biology. Which statement about the enzyme is not correct? So are you saying that all right, let me slow it down a little bit. Which statement about the enzyme is not correct? Are reverse transcriptions unique to retroviruses? Are reverse transcriptions is packaged in a retrovirus particle? The RT protein has the RNase H activity. The name of the enzyme comes from its ability to reverse inflow of genetic information. Might have bridged the RNA world with the DNA world. He said, I dove off the Empire State Building. Think about it. Are RTs unique with retroviruses? If that's a true statement, then move to the next one. Did we say that in the earlier reading? Did we say the second one, third one, fourth one, fifth one?
Question, so I'll find it. Go back to this baby head. What do we get? Which is not correct. RT is unique to retroviruses. It's not. It's in cells.
RT is unique to retroviruses. No, they're not. They're not unique. As he just told you there, they are found in the cell. All the other things he talked about. He talked about all the other things. But he never said A. And I never read A in the first in the first page of this book. Now, I know some people don't actually have the book. That's why I read the first page, first 2 pages. Because it wasn't in the book. But let's continue.
It's packaged in the particle for sure. It has RNase H activity. It reverses the flow, and it bridges the worlds for sure. All of that is correct. So let's resume our discussion. Right, so we're going to talk about reverse transcription now, how it works. So here is the virus particle. Remember, there are 2 viral RNAs.
I'm on page 328 in the 5th edition.
Diploid, if you will.
Yep, the 5th edition. It's under essential components. It says genomic RNA. Let me slow them down. All right. So on page 328, chapter 10, in the book that I have, I don't know if Chavis is still paying attention and someone's using the PDF that he put in the link, which is a different edition, a later edition, which is a different pages. And I don't know if the information matches. However, dealing with essential components, this is a 10-2 cat, and it says here that retrovirus particles contain 2 copies of the RNA genome held together by multiple regions of base pairings. When purified from virus particles, this RNA sediment at 70S, as expected for a dimmer of 35S, and I guess we'll get to that, genomes. Partial denaturation, if I said that correct, an electron microscope analysis of 70S RNA show that the most stable pairing is via sequences located near the 5' ends of two genomes. Sequence interactions that promote the dimerization have been identified in HIV-1 RNA. The 70S RNA also includes two molecules of a specific cellular transfer RNA, or what we know it as tRNA, which serves as a primer for the initiation of reverse transcription discussed in the following section. So despite the fact that two RNA genomes are encapsulated, only one copy of the integrated retroviral DNA is typically detected after infection by a single particle. Retroviral viruses particles are said, therefore, to be pseudo-diploid. Remember that. Retrovirus particles are said, therefore, to be pseudo-diploid. The availability of two RNA templates could help retrovirus survive extensive damage to their genomes. At least parts of both genomes can be and typically are used as templates during reverse transcription, accounting for high rates of genetic recombination in the viruses. Presumably, being able to patch together one complete DNA copy from two random interrupted or mutated genomes would provide survival value. Nevertheless, genetic experiments have shown that the use of two RNA templates is not an essential feature of the reverse transcription process. All of the known steps in reverse transcription can take place in a single genome. Now, like the genomes of strand RNA viruses, a retroviral genome is coded along its length by a viral nucleocapsid protein, which is labeled NC nucleocapsid, with approximately one molecule for every 10 nucleotides. This small base protein can bind both RNA and DNA and promote annual nucleic acids. Biochemical experiments suggest that NC, again, nucleocapsids, may facilitate template exchanges and function in reverse transcription to enhance positivity, the ability to continue synthesis without disassociating from the template, the ability of NC to organize RNA genomes within the virus particle and to facilitate reverse transcription within the infected cell may account for some of the differences in efficacy observed when comparing reactions reconstituted in vitro with those that take place in infected cells. Then he's going to mention primer tRNA when he gets there. But right now we know RT is reverse transcription per virus particle. He's explaining RNA coded with NC. What is NC? Nucleocapsid protein. And the dose usage is 50 to 100 molecules. Reverse transcription per virus particle. In the book, he mentions it under primer tRNA. He says, in addition to viral genome, retrovirus particles contain a collection of cellular RNAs. These include 100 copies of non-random samples of tRNA. In his description below, he also mentions the diploid retroviral genome includes the following 5' to 3. When he's talking about that 5 and 3, that's the DNA, double-stranded DNA. And then he gets into the loop. So let's go. Let's see what you got for us, Vince.
So not just one as shown in this picture. Here's the viral RNA stretched out as a line on the left. It's capped at the 5' end. So it's a messenger RNA. It's 3' polyadenylated. They are the GAG, pol, and envelope coding regions. And look at this. There's some tRNAs hybridized to it. And so, yeah, those tRNAs are cellular tRNAs. They're in the virus particle. And they're going to be the primer for reverse transcription. So the red is the primer binding site. Let's go.
Okay, so the red is the primer binding site. The tRNAs, I need him to say that again.
Coated with nucleocapsid protein, and there are between 50 and 100 molecules of reverse transcriptase per particle, so not just one as shown in this picture. Here's the viral RNA stretched out as a line on the left. It's capped at the 5' end, so it's a messenger RNA. It's 3' polyadenylated. They are the GAG.
Okay. I'm yawning, my bad. The tRNA, I got it. Okay, so however, one particle tRNA molecule is critical. It serves as a primer for the initiation of reverse transcription. The tRNA serves as a primer for the initiation of reverse transcription. And then he says the red is a reflective of the infection site or injection site or whatever he said. But all right, so now we know the tRNA serves as the primer for the initiation of reverse transcription.
Here's the viral RNA stretched out as a line on the left. It's capped at the 5' end, so it's a messenger RNA. It's 3' polyadenylated. They are the GAG, pol, and envelope coding regions. And look at this. There's some tRNAs hybridized to it. And so, yeah, those tRNAs are cellular tRNAs. They're in the virus particle. And they're going to be the primer for reverse transcription.
Now, there's another word that we need to pay attention to here, and that's PBS. PBS is the primer binding site. All right. The primer tRNA is partially unwound and hydrogen bonded to complementary sequences near the 5' end of each RNA genome in the region called the primer binding site. The RTs of all retroviruses studied to date are primed by only a few classes of cellular tRNAs. Most mammalia retrovirus RTs rely on tRNA. For this function, and the relevant primer RNAs are packaged selectively into virus particles.
So the red is the primer binding site. Let's go below here and blow up just this region where the tRNA is binding. So here's the 5' end of the viral RNA. Here's the primer binding site in red. So you can see this area is highly structured stem loops. And the primer binding site is where the tRNA from the cell hybridizes. So it's complementary to a cellular tRNA. On the right here is a depiction of how the two RNAs are held together. So that one of these stem loops, stem loop 1, has a sequence here which can bind, because it's inverted complementary, to the same sequence on another stem loop 1. So here you go. These two sequences can base pair, so that holds the two RNAs together in the virus particle. Why are there two? Most viruses have one piece of genetic information in the particle, right? Well, we're not sure, but here's what we think. We think this having two RNAs makes retroviruses relatively resistant to ultraviolet light and ionizing radiation, both of which cause mutations in nucleic acid. And so if you have two copies of all the genes, you can do recombination during reverse transcription to build a functional genome.
All right, so this is on page 333 of the 5th edition book, and he's using copy choice and strand displacement synthesis is what he's referring to here. So we should probably see two slides. One slide dealing with what he's talking about, the RNA dimmer, and it's just basically, my bad, hold on. Yeah, he's just talking about reverse transcription promotes recombination. And he doesn't really say too much, but he does have a model that emphasizes these things. And let me see, figure 10.8, 10.6, 10.7, 10, where you at? 10.4, 10.8. Okay, so it says here, and I'll read this in here. It says exchange of single-stranded ends from the DNA template to another during the plus strand synthesis can also lead to recombination via a mechanism known as strand displacement synthesis. In figure 10.8, he explains it. Virtually all retroviral recombination occurs between co-encapsulated genomes at the time of reverse transcription. In a copy choice model A, which is what he's showing on the screen, genetic recombination occurs during RNA direct negative strand DNA synthesis. This means mechanisms predict that a homoduplex DNA product is formed as a recombined negative strand of DNA is copied to a form of a positive complementary strand. In the strand displacement synthesis, which is model D, which I think that he's going to get into, he's going to talk about genetic recombination occurs at the plus strand of the DNA synthesis initiated at the negative strand site. So what we're looking at here, remember, when he shows green and blue or whatever, he's really representing those ends of the DNA. And this is the negative is the strand of DNA synthesis that starts on one genome and switches to the second at a breaking point to pause site or random location. So that's what you're seeing in the example below when he talks about that copy choice situation. All right, let's go.
So, for example, we have the two RNAs here on top and bottom, and one of the RNAs has a mutation here in red, little a. So that would inactivate infectivity. But what happens during reverse transcription is actually the enzyme goes back and forth randomly between the two RNAs and copies bits of both. And so at random, it's going to copy the wild type gene in A without the mutation and make an infectious particle. Okay, so that's called copy choice when the enzyme goes back and forth between the two templates. So we think having two RNAs makes it more resistant to mutation. Again, a close look at the tRNA primer now. Here's the tRNA bound to the whole genome. We're now focusing in on the very left end of cap.
This is going back to page, we're going back now to page 329. 329 has the diagram, at the bottom. And what you see, he's pointing his arrow at the DNA strand site, 5' mRNA right there. It has that, and he's going to walk through. This talks about the primer tRNA binding into a retroviral RNA genome.
Is here, there's the primer binding site. The tRNA is hybridized to it in the virus particle. And the way this works is the RNA, which is shown here, is extensively base paired, this big stem loop. And the tRNA is complementary to some sequences here, and it insinuates itself into this stem loop. So this is the tRNA in like olive green, and it hybridizes to the viral RNA in this manner by kind of inserting into this stem loop. And so that's the way it's present in the virus particle. So the primer binding site right there. And then I just want to point out two other sequences in this 5' end of the genome that are going to be very important in a moment. All right. U5 and R. These are just names for sequences. It's a stretch of nucleotides that have been called R and U5.
Did y'all catch that? The 5' primer site. He's telling you that's the 5' primer site. And then you see the other strand of DNA that'll represent the 3' primer site. But now he's going with the R and the U5 and all of that, and he's about to break it down. So let him start off.
In a moment. All right. U5 and R. These are just names for sequences. It's a stretch of nucleotides that have been called R and U5. And we're going to come back to that in a minute. So we have R, U5, and PBS. All right, so let's go through the process of reverse transcription.
Page 331. He doesn't have the top diagram, but the ones at the bottom, he does have on 331 and 332 in the chapter. So he goes back and forth in this chapter pretty heavy, more so than he did the last one.
This is really remarkable, but it's also Baroque, which means it's fancier than it needs to be. Okay. Baroque furniture, Baroque art, and so forth. So DNA synthesis is entirely cytoplasmic. The virus binds its receptor, the nucleocapsid is put in the cytosol, and it undergoes reverse transcription to make a double-stranded DNA right there in the cytoplasm. So let's see how reverse transcription occurs. Here's the viral RNA, one copy we're going to look at for simplicity. The primer for reverse transcription is the tRNA hybridized to the PBS. So RT comes in and makes a DNA complementary to the RNA. This is a short piece of RNA, so it stops. It reaches the end. And you may say, why would you put the primer near the wrong end, the 5' end, when you can't go any further? You'll see. So now we have, and RNase H is chewing up the RNA template as the enzyme makes a DNA copy. That's what those dotted green things mean, okay? So now we have a piece of DNA attached to the tRNA. And remember now, because the RNA had RU5, now the DNA is RU5. Next thing that happens is the polymerase then goes to the 3' end of the RNA and continues copying. And that's because the 3' end of the genome has U3R, so little r and little U3. It's the same little r as on the 5' end. But now we've made a big R, which is a complement in the form of DNA. So the big R can hybridize with the little r at the 3' end of the RNA. This piece of DNA is called strong stop DNA. It's just because you make a lot of it and it's short. So that big R hybridizes with the little r. That's called a template transfer or a template jump. And the polymerase is going to continue, as you can see in the next slide. So that's a first template exchange because we're going from the 5' end to the 3' end. Big R anneals with little r, and the polymerase continues to make the DNA, and the RNA is chopped up, except this little bit here, PPT, polypurine tract. Purines are certain kinds of bases, right? It's not digested by RNase H because it's going to be used as a primer, as you'll see here, for the second strand of DNA. So the first strand goes all the way around. That's the plus. That's the minus strand, actually, because we're copying a plus RNA. This is not the right coloring. You know, plus DNA is dark red. It's been this way for so many years. Anyway, the first strand goes all the way around, chewing up the RNA. And then, before the first strand is even done, the second strand synthesis begins using PPT as a primer. You can see by that light-colored blue there. All right. Next step. So now the synthesis continues here. It's going to reach the tRNA. It actually copies the sequence in the tRNA. All right. So that little second strand of DNA, it copies the entire first strand, and then it goes into the primer binding site. And so this emphasizes the point that this PBS sequence is copied twice, once from the RNA genome, which we have done, and once from the tRNA primer. So now the tRNA is removed by RNase H. It removes the tRNA and it removes the polypurine primer. And then DNA synthesis continues. So now we have a complete first strand and a partial second strand. The second strand copied the primer binding site from the tRNA, right? And so now that second strand can hybridize with the PBS, and the enzyme can continue around the circle and make a complete double-stranded DNA. So this is where we left off. The enzyme continues around, makes a complete double-stranded copy. And so what you see has happened is that we have copied and restored a PBS and a PPT, and we now have at each end U3RU5, U3RU5. And we stretch it out here. Now we have what we call the two LTRs, the two long terminal repeats, because these are repeated sequences. U3RU5, PBS is right next to it. And at the other end, U3RU5 and PPT is right in front of it. You already know what PBS and PPT do. PBS, primer binding site, PPT, polypurine tract. And the functions of those we've gone over. Now, what has just happened in this Baroque or Rococo process, as MK has called it, I guess either would work. Remember, the RNA only has RU5 at the 5' end and RU3 at the 3' end. The process of reverse transcription, but those jumps and everything, has made a double-stranded DNA copy where we now have U3RU5 at both ends. And that's important, as you will see in a moment, because transcription starts at R and ends at R. So you don't transcribe U5 or U3. So the RNA reverse transcription is built to restore it without them being there. That's why you have to have these weird jumps and so forth. So I made an animation of this, which you might want to check out, that sort of clarifies all of this in one short video. Okay, so you might check that out. So again, the RNA lacks the U sequences of the LTRs, and those are restored at both ends. The RNA only has one U3 and one U5. They're duplicated by reverse transcription. Okay, let's do a quiz.
Y'all can't get this wrong, bro. Can't get this wrong, bro. Which of the following steps occur during reverse transcription or retroviral genomic RNA? A, prime of the negative DNA synthesis by tRNA. 2, template exchanges, degradation of viral RNA, generation of 2 LTs, or all the above. Hell yeah, this class hard. But it's okay though, because this is our last question, and then we're gonna deal with the rest of it tomorrow. But I do want to say to everybody listening right now, I'm about to go live on the Massey Warrior Clan. I shouldn't do this on the Massey, not with all the stuff. I'm gonna do a different show. I need to do a different show. I gotta do something real quick. All right. I got to do something after this. Everybody answer? Come on now. All right, so here we go.
Okay, how did we do? Of course, it's all of the above. Yes, everything is.
All the above. With the following steps occurring during reverse transcription of retroviral genomic RNA, it does all the above. We heard all of those things. Generations of 2 LTRs, degradation at the viral RNA site, RNS-H. You heard me read that earlier. 2 template exchanges, priming of the negative DNA synthesis. We making it a little bit more complicated than we will because we're not digesting some of the vernacular. I mean, some of the terminology. I said vernacular. Dumb smack. But you know, let's see the explanation.
Occurring. Priming of minus by tRNA, 2 template exchanges, degradation of the RNA, and generation of 2 LTRs. It's all good. It's all correct. So what we have done, we have made this DNA, and remember, it's got 2 LTRs, and in the middle, the rest of the viral DNA. And what's going to happen next? It's going to integrate into the host DNA, which is shown here in purple.
All right, hold on. I don't want to go too far there. Give me a second. Let's stop the show right here. We don't want to cloud our minds too much. We want to make it digestible for us to be able to go back and learn from it. Yeah, well, I got to do a separate show. But yeah, this chapter 10 was pretty difficult. I think that we just have to continue to work, you know, be more influential with it and deal with it. And then we'll go from there and we'll try to see what we all can do. So give me about a couple of minutes. Actually, yeah, I ain't tight right here. I got Nzinga video stuff still open. Yeah, I can't end that. Was doing good. But I gotta do this one. Dude, hold on, y'all. Just give me a second. Actually, you know what? I ain't gotta give me a second, man. Y'all already know what to do, man. Like the show, share the show, tell a friend. Let's get the crowd up. I mean, most people ain't gonna build like we build, but it is what it is.